Hi class, in this video we're going to go over muscles. Okay, so unit 7. So there's 3 different types of muscles, if you remember from histology. We have cardiac muscle, which surrounds the heart, smooth muscle, which surrounds other organs, and skeletal muscle, which is attached to mainly bones and helps us to move our body. So first we'll get into the functions of muscular tissue. So that first function is going to be movement, right? Muscle pulls on bone to move our body in different ways. Muscle also helps to keep us stabilized. So there's always short muscle contractions helping to keep us stable, okay? So that we don't fall over while we're walking, we can keep our balance, things like that, okay? Muscle also generates some heat, okay? So muscle generates heat. Okay, so those are the major functions of muscular tissue. Some properties of muscular tissue: it is electrically excitable. So remember when we talked about cells, our neurons and our muscle tissue are electrically excitable. So that means they can get signals from nerves in this case and have some sort of effect based on those signals, or some sort of response based on those signals, okay? They are contractile and extensible, so they can shorten and lengthen. And they're also elastic. They exhibit elasticity. So that means that they can go back to their regular shape. Okay, so they can stretch or contract, but then go back to their regular relaxation size. Okay. Okay, muscle tissue components. So we have our actual muscle, like let's take the bicep, right? That's a muscle everyone knows, okay? So the actual muscle, the bicep, has a covering on the outside, okay? So tissue covering. This is known as the epimysium, okay? Epimysium. Then our next unit within our larger muscle, so let's kind of draw this out here, okay? So we have our muscle here, covering this would be epimysium. And then within this muscle, we have what's known as fascicles. So it's like these bundles, okay? So these are fascicles, and these are covered by a covering known as paramecium. Okay, so I'll draw a little arrow in here. This is paramecium. Okay, paramecium. Okay, so epimysium on the outside of the actual muscle, paramecium covering the fascicles, okay? Then within the fascicles, we have our muscle fiber, okay? So you would have, I'm just gonna draw it in one of these fascicles, but you would have these muscle fibers. And these are covered by endomysium. Okay, so I'll draw like an arrow into here, endomysium. Okay, epimysium covering the outside, covering the fascicles would be paramecium, and then covering the fibers is endomysium, okay? Then we have our myofibrils. So that'll be our smallest unit. So coming out of here would be a myofibril. So pretend like it's coming out at you. Okay, so this would be a myofibril, which we're going to talk about in detail. Okay, myofibril. So you'd have tons of myofibrils coming out of these muscle fibers here. Okay, so you do need to know about those coverings there. Okay. Okay, so let me erase this and then we'll talk about the microscopic anatomy. Okay. So for our muscle, the outside is known as the sarcolemma. Okay, so we have this sarcolemma covering, just draw it like this, and we have these things that kind of come in here, okay? These are T-tubules or transverse tubules. So So this here are T tubules. This here is sarcolemma. Okay. And then we have sarcoplasmic reticulum, which is similar to the endoplasmic reticulum of a regular cell. So you'd have here, I'll just draw SR, but this is sarcoplasmic reticulum. And let me just erase this here. Here we'll draw right here. This is a T tubule. And here we'd have another sarcoplasmic reticulum. So they're on either side of the T tubules. Okay, sarcoplasmic reticulum. And then within there we have our myofibrils. Okay, so those are the smallest units, right, found within our muscle fibers. Okay, so they're like these long cylinder regions. And we'd have this throughout the muscle fiber. Okay, now there's lots of different components of that myofibril. So we're going to talk about those. Also within this muscle cell, you have different things like mitochondria, myoglobin, okay, glycogen. Okay, so you'd have other things, nuclei floating through here. So it's like a different type of cell, okay? So let's zoom in on this here and draw out a sarcomere. So let's just erase this for now. We're probably gonna draw something like similar to this again, but let's erase it for now, okay? So let's draw out our sarcomere, okay? So it'd be like a cylinder. I'll try to draw it out as best I can. Okay, so there we have our myofibril, okay? We have here this area is known as our Z disc, and we'll go into detail on what all these things kind of do. Most of these are just going to be proteins. Z disc. So from Z disc to Z disc is your sarcomere. Okay. Then from our Z disc we have here. Let me use a different color and a thinner one here. We have these thin fibers. Okay. So we'd have these on either sides of our Z disc, and they'd be going all the way down to the bottom, of course. Okay. So we have those thin filaments. And then between those, we have our thick filament. Okay, so it's a lot thicker of a protein here. Okay, so these are thin filaments here and then thick filaments, okay? And these thin filaments would actually be a little bit longer. They would go maybe to about here. We'll just draw a few of them longer here. Just erase this. Okay. Okay, so it's our basic sarcomere here. So this is our myofibril. There's lots of other proteins within here as well that we're gonna go over, okay? But let's just talk about kind of what's going on here first, okay? Let's draw actually our titin filament as well. So our titin filament, let's draw in a different color. Use blue, okay? Our titin filament, it's another protein within here that's between the Z discs, and it's kind of twirly here, and then it just keeps going. And then it gets kind of twirly again over here. Okay, let's draw a few of these. Again, this is a titin filament. So within the center here of the sarcomere, we have another thing going on known as the M line. So I'm just going to draw everything out, name everything, and then we'll go over their actual functions here. Okay. So this here would be our M line. It's going to go all the way down, so let's just keep drawing this one since it's kind of easy to draw here. Okay, so it's our M line. So this here is our M line. This in blue here is our titin. Then we have our thick filament. And we have our thin filaments. Okay, so thin filaments. Okay, and then we have our Z discs. Okay. So where these things overlap, our thick and thin filaments overlap, is known as the zone of overlap. So let's say like from here to here, that there's our zone of overlap. Okay, zone of overlap. Then we have our H zone, which is just where we have thick filament and no thin filament. So this here would be our H zone. Okay. Then we have our A band. That's the whole length of the thick filament. Okay, whole length of the thick filament is the A band. A band. Okay, and then we have our I band, which is just where we have thin filament and no thick filament. So right here, let's say, this would be the I band. Okay. Okay. So let's go over all these different parts. So the Z disc are, again, from Z disc to Z disc is one sarcomere, and then it would just keep repeating as you go on, but I don't have a lot of time to draw all of these, right? So this is one sarcomere, and then all these things just keep repeating on down the line, down the myofibril, okay? Our A band, again, this is the middle part, and it extends the entire length of our thick filament here, okay? I band, this is gonna be under the microscope, it'll look lighter colored because this is just where we have thin filament, no thick filament right there, okay? H zone, this is just in the middle of our A band there, okay? And it contains only thick filament and no thin filament. So I band, only thin filament, H zone, only thick filament, okay? Zone of overlap is where they overlap, so I'd have both thick and thin filament. And this M line here is made up of proteins, okay, and it's in the very center of the H zone there, okay? And it just helps to hold the thick filaments together at the very center of each sarcomere, okay, the M line. So what are thick and thin filaments? Let's get into that, okay? So thick filaments are actually myosin, okay? Thin filaments are actin, okay? These are proteins, okay? So thick filaments are made up of myosin, thin filaments are made up of actin, okay? Now, our thick filaments are going to be these structures that are twirled together. They're just kind of twirled up. I'm not great at drawing this. Twirled up together here, and they have two heads. Okay, so two heads here. And then on each head, you have an actin binding site. So this here would be the actin binding site. Okay. Our actin are also two strands, but they have like these bulbs that make them up. Okay, then they'd have more bulbs kind of crossing not the best artist in the world, but that's as good as I can do there. And these have little myosin binding sites. Okay, so they have little dots where the myosin can bind to, the myosin heads would bind to these myosin binding sites. Okay, so the myosin heads have actin binding sites, the actin have myosin binding sites. Okay, they also have these two other proteins, troponin and tropomyosin. So let's draw them in a different color to make this more easily seen. Okay, so our troponin we'll draw in green here. So it's gonna be something like this, these three little balls representing a protein. Let's draw a few of them just to show you. So this is our troponin, okay, troponin in green here. Okay, troponin. And then in our red color, we'll draw tropomyosin. It's these long strands that cover those actin binding sites. Okay, tropomyosin. Okay, and we're going to get into what happens here very soon. Okay, so then we have different other structural proteins. So I talked about the titin here that I drew in blue, right? That kind of spans across the sarcomere. Okay, so titin connects the Z disc to the M line in the center of the sarcomere. Okay, so it helps to stabilize the whole structure, the whole sarcomere. Okay, we have other proteins, so alpha actinin. This is a structural protein on the Z disc, and it attaches the actin molecules to the titin molecules. So it attaches the thin filament to the titin. That's alpha actinin. Myomycin, this is the protein that forms this M line here, myomycin. Okay, so it binds to the titin molecule and connects the thick filaments to each other. So the thick filaments that are kind of adjacent here, it connects them. Okay. Nebulin, this is a protein that wraps around each thin filament, each actin filament. Okay, and it helps to anchor the thin filaments to the Z disc. So there's lots of structural proteins that help to maintain the structure of the sarcomere. And then finally we have dystrophin. So this links the thin filament to other membrane proteins in the sarcolemma. Okay, and it also helps to connect it to the matrix that surrounds the fiber. So it helps to connect it to the matrix around the fiber here. Okay, so these proteins, just know in general what they are, what they do, but in general they help to keep the structure of the sarcomere. Okay. Okay, so now we're going to get into the sliding filament mechanism. So this is how a muscle actually contracts, the sliding filament mechanism. Okay, so what happens is we see how, I'm going to show you here, give me one sec. Okay, so we see how the thick filament and thin filament are right near each other, right? So what happens is these myosin heads bind to the actin, the myosin binding sites on actin, and they pull the thin filaments in, okay? So they cause the thin filaments to slide inward like this, okay? So when that happens, these Z discs are gonna move closer to each other, and the sarcomere, the whole sarcomere itself is going to shorten. Okay. And we see how these, it's kind of hard to tell from my drawing, but you see how these thin filaments don't go all the way to the M line here. If there's a full contraction, they can though, because again, these thick filaments pull on the thin filaments, or the actin, the myosin pulls on the actin and pulls it toward the M line in the center. So say the M line is the center or my midline, right? So the myosin binding heads bind to the myosin binding sites on the actin and pull them inward toward the M line. The whole sarcomere shortens, so Z discs will come closer to each other, okay? So because of all those structural proteins we just discussed, there's a force throughout the entire muscle that results in the whole muscle contracting, because all those proteins are also helping to hold together the next sarcomeres on the sides here. So it'll cause a contraction of the entire muscle throughout the myofibril, okay? So it's kind of like a chain reaction. Okay, so we have all these different bands and zones going on here, okay? So what shortens and what stays the same when we're talking about a muscle contraction, the sliding filament mechanism, okay? All right, so we have our H zone. This is where we just have thick filament. If we have a full contraction here where we pull the thin filament over, this H zone can shorten or disappear depending on the strength of that contraction, okay? So the H zone is going to shorten and or disappear, okay? Our I band is just where we have thin filament. So this can also shorten or disappear, right? If it's being pulled in, it's being pulled over the thick filament. So let's say our Z band is going to kind of move over here, okay? And then we won't have any area of just thin filament. It'll all be overlapping. So our zone of overlap will be the entire thing instead of just this little area here and here, okay? But what happens to our A band here? Okay, so our A band is the entire length of the thick filament, okay? So our A band, our thick filament doesn't change in size. It pulls the thin filament over it. So it never changes in length itself, right? I myself, if I'm pulling something over me, I don't change in width, right? The sarcomere changes in width. So the A band never changes its size. It's not going to shorten or disappear, okay? It's just gonna stay its same length, okay? So again, the H zone and I band, the areas where we just have thick filament and just have thin filament, of course, are going to disappear during a full contraction, but our A band is going to stay the same length. It's not going to change, okay? Okay, now let's talk about what causes this contraction to occur. So it's known as excitation-contraction coupling. Okay, so those are the events that send what's known as an action potential. So an action potential is a signal that tells the muscle to contract. So that's of course going to be a nerve telling the muscle to contract for whatever reason.